What's up everybody? So in this video we're going to be looking at this crazy cute tiny tiger, but also we're going to be looking at DNA replication. This is a huge topic for IB biology, okay? So I'm going to try and make this as easy as possible. But I must emphasize, to understand this video as good as possible, you should watch my video or any video of anybody who explained the DNA structure first, okay? Otherwise it's going to be like learning how a car works without having ever seen how a car structure is, okay? You need to learn the structure before you can learn any kind of function like this, okay? So anyways, we have here Brad Pitt, okay? We got Brad Pitt here, and he's gonna represent our regular human. Even though he's pretty famous, he's still a regular human, okay? So when we look real close here at Brad Pitt, we know he is made up of what? He's made up of trillions of cells, okay? And inside, he has a bunch of organelles, right? Each cell has a bunch of organelles, and the organelle we're going to care about in this video is the nucleus, okay? What exactly does the nucleus have? The nucleus has DNA, okay? Deoxyribonucleic acid, DNA. This is your genetic code. This is what makes you who you are, right? This is like the instruction manual of your body, okay? Your cells will read your DNA and make sure to become exactly what the DNA says. It's like the instruction manual, okay? Now, my question is, why are we talking about DNA replication? What does DNA replication mean? Replication means to make more of, to duplicate, right? So why on earth would we want to duplicate our DNA? Why? We want to replicate our DNA when our cell divides, okay? What do I mean by that? Look, if we have a cell, okay, and it divides to become two cells, right? It divides. If our cell doesn't duplicate the DNA or duplicate the organelles inside, then these two cells will have half of what the original cell had, okay? It will have half of everything. And that means the cell is not a proper cell. It needs to have everything proper. So before this cell divides, it needs to duplicate everything. Duplicate DNA, duplicate organelles, so that when it divides, the two baby cells will have all of the DNA it needs, okay? It won't have half the DNA, it will have all of it, okay? So it will be exactly the same as the original cell, okay? That's why duplication is so important, replication, okay? So why do we care about the replication? Why do our cells need to divide? Our cells need to divide or get used to dividing for two reasons. One, growing up, okay? The only way you can become from a teeny tiny baby into a human being, into a full-grown adult, is if your cells divide and become more and more so that you can increase in size, right? So that's one reason why. And another one is because of injuries, okay? So say you fall your absolute face off on the concrete and you scrape off all your skin and you form a scab. The only way this can heal is if the cells around it divide to replace these dying cells or these dead cells here, okay? So that's another reason why cell division is so important. Now, let's go here, okay? So now we're looking at a piece of our DNA. So how is the basic concept of how our DNA divides, okay? So I'm going to show you now how it does not divide, okay? How you may think it does, but it really doesn't, okay? This is what I think everyone would think of initially, but this is actually not how it works. So most people would think, okay, you have your original, so you have your piece of DNA and you're going to synthesize a brand new one. So you're going to have now two pieces of DNA. You duplicated this DNA by having the original one and synthesizing a brand new one. So I'm having this one green, not for any particular reason, just to show that it's a new one. Okay, it's a new piece of DNA. So this is not how it works. You're not going to have start off with one DNA and then synthesize a brand new one, and now you have two pieces of identical DNA. Okay, that's not how it works. Okay, so, oh yeah, that's also one important thing I need to say. When you duplicate DNA, you're going to make it the exact same as the original DNA. So you're going to synthesize the exact same DNA, because if you don't do that, then your cells will have the wrong code, the wrong information, and you will not function properly. Okay, you need to duplicate exactly your DNA, the same way it was before. Okay, so that's why these two pieces of DNA are exactly the same. If you look at the bases, okay, they match exactly. We got orange, red, orange, red, red, orange, red, orange. Okay, that's how it works. So this is not how it works. So how does it work then? So what's going to happen instead is something slightly more complicated. You're going to have your original piece of DNA, and essentially it's going to get separated. It's going to get opened up like this. And then you're going to synthesize the complementary strand right here to form a new piece of DNA, and they're going to synthesize the complementary strand of this one right here, and you'll have a new piece of DNA. What do I mean by that? This, okay? So we separated our DNA and we synthesized the complementary strand here, a new one, and then we have this other old one, and we synthesized a brand new strand there. Now, these are two pieces of DNA that are exactly identical to the original one, okay? You can compare. It's going to be the exact same. The only difference is, compared to this one, is that in this one we had an old DNA and we had a new DNA completely. Whereas this one I'm showing you here, which is the right method, we're going to have two pieces of DNA, but they're both going to be half old and half new, okay? Because this strand right here, this strand right here, and this strand right here served as a template, okay, for the new strand to be formed. Because this was the template, and then the new strand binded by complementary base pairing to form a new strand. And this is a full-on DNA now. So we call this process semi-conservative. Why? Semi means somewhat, pretty much means somewhat, and conservative means the same, okay, to keep the same. So this process is semi-conservative because we somewhat made a new DNA. We somewhat kept the old DNA because we kept half of each new strand of DNA will have half of the old and half of the new. So it's semi-conservative. It conserves half of the DNA. Each new DNA will conserve half of the old DNA. Okay, it's semi-conservative. This is a very important term I think you need to know. So that's the basic concept, okay? So it doesn't work like this. You don't synthesize a completely new one. You rather do it in a semi-conservative way, okay? Just like this. So how exactly does this happen? How do we separate apart this DNA and then synthesize this new strand onto it? How does that work? So that's exactly the purpose of this video. I'm going to go into all the details you need to know to be able to get full marks on whatever DNA replication question you get, okay? So let's just get into it. So let's pretend here we have our DNA. Okay, we're looking here at Brad Pitt's DNA. Remember, we're using Brad Pitt here. So this is his DNA, and remember, DNA kind of is normally coiled up like this in this helix shape, okay, like this helical structure, right? So all these bases on the inside, remember, these are like the letters, the A, C, T, and G. They are kind of hidden on the inside, okay? They're hard to access. It's hard to like make new DNA when the DNA is coiled up like this, right? So what do? So the logical thing is to uncoil it. So that's exactly what happens. There is something called an enzyme called helicase. Helicase, and it sounds like helix, right? That's because it's going to uncoil this helix. It's a helicase. Any name that ends in an ace is an enzyme, right? So this helicase is going to uncoil it to make it like this. And then it's going to do one more other thing. It's going to separate the two strands because we got one strand here, okay? And they are complementary to each other, right? The green always binds with the blue and the red always binds with the orange. What do I mean by this? Remember, we have four bases: guanine, cytosine, adenine, and thymine. A always binds with T. You can remember it as A and T are always, A for always and T for together. So they will always bind together, and the green and the blue will always bind together, right? This is called complementary base pairing. Now, the helicase is going to basically go here, and it's going to serve like a little scissor. It's going to break these bonds in between these bases. It's going to separate these two strands, okay, like a scissor. And remember, these bonds that hold these two bases together between the two strands is hydrogen bonds. And if you remember from my DNA structure video, hydrogen bonds are quite weak. They're easy to break by this helicase. So this process is pretty easy for helicase to do, okay? So what next? So we managed to open now and see what's going on more clearly. It's much more easy now for the rest of the stuff to happen. So what happens next? So right now we separated this DNA, right? This helicase, I'll bring it back here for you. This helicase cut open the DNA, right? We're about here now. We haven't cut these yet. Now there's going to be another molecule that I'll show you called gyrase or gyrase, however you want to say it. And basically, let me show you. I'll bring it in. It's like it is going to be here, okay? Its job is to basically stabilize the DNA that has not been cut open by helicase yet, okay? Because naturally, DNA wants to coil up like this, okay? So if nothing is stopping it from coiling back up, then it's going to coil back up again. We know helix uncoils it, but something has to stop it from recoiling, okay? And basically, this gyrase, I'll bring in the name, gyrase, is an enzyme, again, because it ends in ase, that is going to help stabilize the DNA strands so that they don't recoil, okay? It's going to stabilize them, and that's going to make it easy for Helicase to just cut them open, okay? So that's what gyrase does. Now, this whole structure here kind of looks like a fork, doesn't it? We have the handle here, and we have the sharp fork pieces coming out, okay? We got a two-edged fork, okay? So we call this whole structure replication fork. This is the whole replication fork. And again, that's another key word you need to know, okay? Because replication is happening here, and it looks like a fork. Very simple. Remember from the DNA structure video that when we have two DNA strands, okay, like this, okay, let me show you here. So say we have these two DNA strands. The one strand will have a 3' end at one end, and then the other strand will be called 5' end instead, okay? So they are anti-parallel, okay? And if you don't know what this means, the 5', 3' thing, again, go watch the structure video. It'll make a lot more sense. So anyways, the one strand will go from 3' end, so it's basically like saying head or toe, and then the other side is the head, okay? So we call the other side the 5' end, 5' end. So if you follow it around, it's going to come here to the 5' end. Now the opposite strand is the exact opposite. So the 5' end will be on this side, and the 3' end will be on the other side. So you can see it's the opposite, okay? And this is going to be important for later in this video. So what's next? We opened it up. We opened up and separated the two strands. Now what exactly is going to happen next? Now we want to synthesize this new strand, right? This is the old strand. We're going to synthesize this new one onto it to form a new piece of DNA. And we want to have this old piece of DNA also have a new piece of DNA. So the total, we duplicated DNA. So what's going to happen is something called, let me show you. Okay. Something called primase. RNA primase is going to come in, okay? And what this little molecule does is something interesting. It's going to grab an RNA nucleotide. So let me show you. It will basically grab an RNA nucleotide. So we say this is a free-floating nucleotide, okay? It's a free nucleotide. And it's going to grab it, okay? And it's going to basically bring it and attach it to its complementary base, okay? We know the green one always goes to the blue one. So it's going to bring it there and attach it, okay? But again, remember, this is an RNA, not a DNA, right? So we're going to have to do something about this later, you'll see. So now, what was the purpose of this? What was the purpose of putting this primer, okay, the primase puts a primer that is RNA. What is the purpose of that? The purpose of that is because this primer acts as like a place for another molecule to go to. It's like a pit stop, okay? It's like an attraction, a meeting point, okay? So it's going to be here. It's going to be like a meeting point for another molecule. This molecule is called DNA polymerase 3, okay? DNA polymerase 3. And again, they named these DNA polymerases in the order of discovery. So they discovered this one third, okay? So that's the only reason why it's called that. So this DNA polymerase is very attracted to this primer. So it's going to go there, okay? It's going to attach right there, okay? Just like this, sort of, okay? Now, once it attaches there, its name is what? Let me move this RNA primase out of the way for now. Now, its name is RNA polymerase. What does that mean? I mean DNA polymerase. Poly means many. Okay, so this is a thing. This is an enzyme because it ends in ase that is going to make many poly. It's going to make a polymer. It's going to make many DNAs. So it's going to synthesize DNA. So it's a DNA polymerase. It's going to polymerase DNA. So what it does is going to go here, bind next to this primer, and drag in some, this time, free nucleotides, okay? These are also free nucleotides because they're free-floating nucleotides, right? We got the backbone and we got the base, okay? And he's going to basically force them to bind by complementary base pairing. But the difference between this one and this molecule is that this is actually a DNA molecule, so it belongs here. So it's going to drag it in, make it bind, just like this, okay? And it's going to continue doing that and match them all. Okay, so here, we did it, okay? Now we synthesized a bit, and what's going to happen next is it's going to move along. So it's going to keep moving along and keep synthesizing 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 all along as these helicase opens up the DNA. Okay? Now, my next question for you is this. It's going to make this even, otherwise it's unsatisfying for those of OCD. So what do we do here? So we noticed this was the 3' end of this old strand, because this here is the 5' end. So this means this new strand, what do we call this side? We call it the 5' end, right? It has to be the opposite. So this is the 5' end. So this DNA polymerase is synthesizing DNA from the 5' end to the 3' end, right? Okay, it's synthesizing it in this direction. So let's put on the rest of the DNA. Here we go. Okay, let's pretend it synthesized all this. Okay, so we synthesized, so DNA is added. This is a very important point. DNA is added, let me fix this, from the 5' end to the 3' end, always, okay, 5 to the 3' end. So we can see that this one on this side, we call this strand, by the way, this strand here, this one, the leading strand, okay? We call this one here the lagging strand. I'll show you how to spell that just now. Now you can see on this leading strand, when we synthesize a new piece of DNA, it is continuous, okay? Because we synthesize from the 5 to the 3' end, and it goes continuously as long as the helicase keeps opening up DNA. It's continuous and smooth and easy, right? You'll see it's not the same on this strand called the lagging strand. And don't worry about any of the processes here. I'm going to write it out. I have a clear image where I write it out in words. Right now I'm just explaining the process so you can see it visually. So don't worry, I'm going to have a nice image where I clearly explain it, okay? So now we're almost done here with this leading strand. Again, this is the leading strand, the strand that is synthesized continuously and easily. So we're almost done. So let's pretend we finished now synthesizing this entire piece, okay? We have a problem here. This is an RNA. It can't be there. It needs to be in DNA. So there's going to be another molecule, okay? We call this one DNA polymerase, but we call it now DNA polymerase 1. Remember this green one here was called DNA polymerase 3, right? So this one is called DNA polymerase 1. And what it does is it goes and takes out this primer, this RNA primer, and replaces it with a proper DNA one. Okay, so here's the DNA one. Let's say, maybe make this a bit thicker. Let's say it takes out this one. Okay, and now it's gonna go pretend it takes it in there, puts the right one in there. Okay, so now we pretty much, let me fix this just for OCD's sake. Okay, that's better. So now we can see that a completely new strand was synthesized. We finished it. They're all DNA pieces. They're all DNA nucleotides, non-RNA nucleotides. And again, they're all matching perfectly, and it's matching the original piece of DNA. So remember, when these two strands were together, it's going to look the same as this strand here. Notice, look, we have green, green, orange, orange, green, green, blue, blue, orange, orange, red, red, so on, right? So this new DNA that's being formed is the exact same as when the DNA was together, as when the original piece of DNA was together. So that's important. It's the exact same DNA we're synthesizing, okay? So don't get confused by that. It's just this is the old strand and this is the new strand being formed, semi-conservative. Okay, so that's it for the leading strand. Let's quickly go on to the lagging strand, because this one is a little bit more painful, okay, unfortunately. Let me move this out of the way. Okay, so now we need our primase again, okay? By the way, this goes on at the same time. It's not like the leading strand is synthesized first and then the lagging strand. They happen at the same time. I'm just explaining one by one for you. Okay, so here we have now, again, a primase, okay? An RNA primase. Where's the name? RNA primase, okay? And it's going to go, same thing. It's going to go take a primer. Let me show you. It's going to take a primer and add it on somewhere here, okay? To allow DNA synthesis to start. So it's going to put it up, let's say here, okay? It puts it on right here, okay? So we had now a primer added, and we know when a primer is added, what does that mean? Let me move this out of the way. Now we can have this DNA polymerase 3 come in and bind to it. Oh, sorry. Let me do this again. Okay, now we can have this DNA polymerase 3 come in, okay, and bind there, right? Because this primase acts as an anchoring point. So when this DNA polymerase binds, why does it synthesize from this one this way instead of from this one this way, like what happened here. The reason why is because I told you that DNA needs to be synthesized from the 5' end to the 3' end. Now, on this strand, this is the 5' end, meaning that here is the 3' end. And this means that the new strand being synthesized, this will be the 3' end, and the other side will be the 5' end, right? So it will be like this, right? The 5' end will be this way, and the 3' end will be on this side. So when this primer is put and the DNA polymerase 3 comes in, it's not going to synthesize. Oh, sorry, it's not going to synthesize from. Wait, what's happening here? It's not going to synthesize from here this way because that's from the 3' end to the 5' end. It's going to go here and synthesize from here this way. Okay, so that's confusing. So you can see that's going to be a little bit more complicated, but let's just go with it. So right now, it's going to take these new nucleotides, going to put them on. So here it goes. It grabs them, synthesize again, complementary base pairing. Okay, so here it goes, and it moves along a bit. Let's say it moves along a bit to finish the job. Okay, now it's going to finish the job there. Okay, so cool. Now we synthesized a piece of DNA. But what now? Because now, what about all this stuff? How is this going to be synthesized? Because we know this helicase is continuously cutting open the DNA to make room for new ones to be synthesized. So what's going to happen with all this? Well, it's very simple. This RNA primase is going to go again, and it's going to put a new primer somewhere else. So let's say he takes this primer, okay, a primer, and he puts it somewhere. Okay, let's say he puts it here. Okay, here he goes. Move this. Okay, so you put it there, you put a new primer, and again, after a primer is put, what does that mean? Now, this DNA polymerase will move around and try and find this new primer and be like, oh, found you, and it's going to stop there, and again, synthesized from this end to that end. So it's going to go, oh, let me fix this OCD stuff. Okay, so it's going to go 5' end to 3' end, always 5' to 3' end. So let's go. He takes it, grabs it, places it, next one. And again, these are DNAs, remember, the primers are RNA. So here we go, satisfying, finished, right? So you can see it's like we're synthesizing bit by bit. So the leading strand was very easy because it's continuous. It's like once a primer is put, you can continuously synthesize continuous until the helicase stops doing its job. But on this side, the lagging strand, it's going to be like bit by bit, okay? So what do we call these small fragments? Because we're synthesizing bit by bit, right? I'll get to the name just now. Okay, so let's say this happened again. I'm just gonna drag it in, okay, just for the sake of time. So let's say it happened again. So we had another fragment. Sorry. Okay, cool. Okay, so just pretend these are all tightly together, okay? So now we have, you can see we did this now 3 times. So another primer was added by RNA primase, and DNA polymerase came in, synthesizing from the 5' end to the 3' end until it reached the previous primer and stopped. Okay, so what do we call these little bits and pieces? Okay, we're synthesizing bits and pieces of DNA. We call these little bits and pieces Okazaki fragments. So this is one Okazaki fragment, this is another Okazaki fragment, this is another Okazaki fragment. So we didn't have this Okazaki fragment on this leading strand here, only on the lagging strand, okay? So, almost done here. So what happens now is logical. So this is going to continue happening, the same process, okay, until the DNA is completely replicated. But, let me move this out of the way. Oh, sorry. So now, what's next? So now we have all these primers and we need to remove and replace them. So what molecule do we need? We need DNA polymerase, okay, DNA polymerase, same molecule as before. So it comes in, DNA polymerase will go and grab these individual primers, take them off and replace them. So let's say it's going to take off this one. I'm not going to really do it because it's going to take some time, but I'll show you one example. So it's going to basically take them off. Take them off. Let me see if I have give me a second. Yeah, here I got one. So it's going to take them off and replace them, just like this. Okay. Okay, so it takes them off and replaces them. Now what happens is, notice how there's a big gap in between. Okay, so it's going to take off these RNA primers and replace them with DNA primers. Okay, I mean DNA pieces, nucleotides. But now there's this small gaps in between. So we need another enzyme. Which enzyme is that? We need an enzyme called ligase. Okay, here we go. I didn't replace this one, but pretend that one's replaced. So now there's going to be another enzyme called ligase, and it's going to go over this, okay, ligase, and it's going to seal all these pieces together. It's going to put this backbone, because we call this the backbone, okay? It's going to go there and seal all these backbone together, okay? Make them connect to each other, okay? So that's what happens. So it's basically what it does. It's going to be hard to show here, but essentially does this, okay? It fixes these gaps in between, okay? So that they're all connected strong and properly. Okay, that's basically what ligase does. Okay, so now we're done with the lagging strand and the leading strand. Okay, that's it. That's the entire process of replication. Now, after the DNA is synthesized, it's going to go, because the gyrase is not here, right? The gyrase helps to prevent it from recoiling. What's going to happen after it's synthesized, this DNA, is it's going to recoil. So you can see here, I'm dragging this over, the DNA will start recoiling, okay, recoiling back to form the structure of normal DNA. Same here, okay, the DNA is going to recoil, okay, slowly and form DNA again. So that's it that you need to know. That's all it is, okay. So right here on the next page, I'll quickly show you a word form of this just so it's a bit more clear. So first steps first, DNA double helix is unwound and separated using helicase, forming the replication fork, right? We know that. Helicase unwinds it, separates the DNA by breaking these hydrogen bonds, forming this replication fork. Gyrase, next one. Gyrase will reduce the strain to prevent supercoiling, right? It's going to prevent these DNA here from forming a helix so that the helicase can do its job. Next, this part you don't need to include in your answer. It's not an IB thing. I just have it here. These strands that are formed when separated, they're very unstable. So there's a molecule called SSBP that can stabilize them, but you don't need to know about this. So you can skip step 3. It's not required in the IB, okay? But then then step 4 will happen. But depending, this step 4 will be different depending on the leading strand or the lagging strand. So again, the leading strand was easy. That's this strand here, right? What's going to happen is the RNA primase will add a primer, and then the DNA polymerase 3 will bind to the primer and add DNTPs. That's just a way of writing nucleotides, DNA nucleotides. So it's going to add these DNA nucleotides to the third end of the primer. What do I mean by that? It's going to add it to this third end, but it's going to synthesize it from the 5' to 3' end. Okay, always 5' to 3'. Okay, so then after that, after synthesizing 5' to 3', DNA polymerase 1 will remove the primers and replace it with DNA nucleotides. In the meanwhile, the lagging strand, which is discontinuous, right, we have to make these small fragments, goes basically the same, except we synthesize still from the 5' to 3' ends, but this makes us form these Okazaki fragments. And again, DNA polymerase will remove these primers and replace them with DNTPs, and then it has one extra step, because there are these small gaps in between the Okazaki fragments, right, these Okazaki fragments, then we need an enzyme called ligase, which will seal up these little breaks in between the Okazaki fragments. And then lastly, the DNA will recoil, okay? And now we basically form 2 separate DNA strands, okay? So once we go through the entire DNA, we'll have 2 completely separate identical DNAs, okay? That is DNA replication for you. Here, just for you, I picked out the longest IB question you can find on DNA replication. So you can notice here, here's the question and here's the answer. Okay, there's so many different, you only need 8 marks, but look how many answers you can give. 1, 2, 3, 4, 5, 6, 7, 8. You can get a total of like 12 marks, okay? So you only need to write 8 of these to get 8 marks. And you can, if you can read carefully here, and you'll see it's exactly what I taught you. So read it carefully and look at the video and see that it matches, and that's all you need to know. So I hope this video was useful, and let me know if there's any problem or anything that's unclear in the comments. I'm gladly willing to help you.